In this video, we are going to discuss about Kingdom Monera. The term Monera was coined by Ernst Haeckel. It was Herbert F. Copeland added Monera as a separate kingdom. These days, it is also called as prokaryotic. Let's discuss the general characteristics of Monera. Monerans are unicellular and prokaryotic. We call them prokaryotes because membrane-bound cell organelles are absent and their genetic material is naked DNA, which is otherwise called as nucleoid or genophore. Ribosomes and simple chromatophores are the only subcellular organelles in the cytoplasm. Chromatophores are the pigment containing structures in bacterial cell. Most of their cell wall is made up of a non-cellulosic material called peptidoglycan. Few exceptions are found like archaebacteria and mycoplasma, which do not possess peptidoglycan in their cell wall. Predominant mode of nutrition observed as heterotrophic, but still few groups are known to be photosynthetic or chemosynthetic. Monera can be widely classified as archaebacteria and eubacteria. Archaebacteria is one of the most primitive group of bacteria. It can also be said as the oldest of living fossils. They are found in most harsh habitats, such as extremophiles. Harsh habitats like extreme salty areas, hot springs, and marshy areas. Their cell wall is adapted to tolerate extreme conditions. As said earlier, peptidoglycan is absent in their wall. The cell membranes are characterized by the presence of branched chain lipids, which make them resistant to heat and low pH. Three main groups of archaebacteria found are methanogens, halophiles, and thermoacidophiles. Methanogens are gram-negative, obligate anaerobic bacteria. We'll discuss about obligate anaerobic bacteria later towards the end of this session. They occur in marshy habitats, swamps, and even in the gut of the ruminants. They are mostly autotrophs, which obtain energy as well as carbon for assimilation from the end products of the decomposition. They can even produce methane from carbon dioxide and are widely used in biogas plants. Halophiles are facultative anaerobic bacteria. They occur in extreme saline or salty conditions. To 35% of salt or sodium chloride must be there in their culture medium. Their intracellular salt concentration is also equally high. The enzymes and ribosomes of halophiles function efficiently only at high intracellular salt concentration. Halophiles have a purple pigmented membrane due to the presence of a pigment named rhodopsin. Rhodopsin utilizes light energy for the metabolic activities. It is actually different from photosynthesis. Rhodopsin actually acts as a proton pump. It captures light energy and used to move protons out of the cell. So this results in the formation of ATP, which is used for further cellular functions. Thermoacidophiles are temperature and acid-loving archaebacteria. They are facultative aerobic bacteria which are found in hot sulfur springs, and they can even survive up to 80 degrees Celsius. They have the capacity to oxidize sulfur to sulfuric acid at high temperature and at high acidity. Some of these bacteria are able to reduce sulfur to hydrogen sulfide under anaerobic conditions. Now coming to the eubacteria. They are true bacteria. Leeuwenhoek was the one who observed small or tiny animal cues in sewage water or saliva and in tooth scum for the first time. And they were later known as bacteria. And Leeuwenhoek is known as the father of bacteriology. Bacteriology stands for the study of bacteria. Louis Pasteur was the one who proposed the germ theory of diseases. It states that certain diseases in plants and animals are caused by bacteria. Bacteria were first cultured by Robert Koch, and the term bacterium was coined by Ehrenberg. These are the simplest and most primitive unicellular organisms, and they are found everywhere: air, water, soil, food, and even inside our body. They are prokaryotes, and hence their primitive type of nucleus is known as nucleoid, and they have the prokaryotic ribosome of 70S. They do lack other cell organelles of complex which are found in the complex cells, and they also possess cell wall. The extrachromosomal structure present in bacteria is known as DNA plasmid. These plasmids are small, circular, double-stranded DNA molecules, and they exist separately from their normal genomic chromosome, and they can replicate independently. If you see to the classification of bacteria based on their shape, they are coccus, which are spherical shaped, bacillus, rod shaped, or even occur in a pair or like a chain, spirillum, spiral or curve shape. Few of them are vibrio or comma shaped, and some of them occur as a filament which looks like the fungal mycelium, and few have stalk, so we call them stalked bacteria. Most of the bacteria are immotile, they cannot move, but few bacteria such as Bacilli and Spirilla can move on their own. They have single polar flagellum at one end of the cell, or they can have clusters or many flagella at one or both ends, as you see in the picture. Based on the nutrition, bacteria can be classified as follows: as autotrophs and heterotrophs. Autotrophs can be further classified as photoautotrophs and chemoautotrophs. Photoautotrophic bacteria have chlorophyll, which is similar to that present in the green plants. For example, cyanobacteria, which is the blue-green algae, which are unicellular, sometimes live in colonies. Their colonies are generally surrounded by a gelatinous sheath. They often form blooms in polluted water bodies, and some of them can fix atmospheric nitrogen with the help of their specialized cells called heterocysts. Example, nostoc, anabina. Chemosynthetic autotrophs oxidize inorganic substances such as nitrates, nitrites, and ammonia, and use the released energy for the ATP production. These autotrophs also help in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophs are most abundant in nature. They are found in different categories such as saprotrophs, symbionts, etc. Parasites are the ones which get their food from living organisms and may cause diseases, whereas saprotrophs obtain their nutrition from dead organic matter. They secrete extracellular enzymes which help in extracellular digestion. The digested solution is absorbed by the saprotrophs. Symbionts exhibit symbiosis. For example, rhizobium that is found in the root nodules of legume fixes atmospheric nitrogen into soluble forms. Another example of symbionts is Escherichia coli in human intestine. They consume food in intestine, releases vitamins like K and B12. Also, they check the growth of harmful putrefying bacteria. Based on respiration, bacteria can be classified as obligate and facultative. Obligate anaerobes are the one who can grow only in the presence of oxygen, whereas facultative aerobes, they can also survive in the absence of oxygen or even the presence of oxygen. Similarly, obligate anaerobes can grow and multiply only in the absence of oxygen, whereas facultative anaerobes can survive in the presence or absence of oxygen. Basically, bacteria reproduces asexually and sometimes they exhibit sexual reproduction. Asexual reproduction is carried out by binary fission, which takes place during favorable conditions where the cells split into two. And they also reproduce by entospore formation. Sexual reproduction is generally absent, but there occurs genetic recombination, that is, bringing together of their genetic material. It occurs by three main methods: by transformation, transduction, or conjugation. Transformation is a process by which exogenous DNA material is transferred into a bacterial cell, as you see in the figure. This transfer of DNA takes place from dead system into the living system. Transduction is a process in which genetic material is transferred from one bacterium to another through bacteriophage. Bacteriophage are the viruses that attack bacteria. Conjugation is the process of transfer of genetic material between two different strains of bacteria. Now, here are a few amusing facts about bacteria. Certain bacteria is used in bioremediation, that is, in the degradation of environmental pollutants. Bacteria is also used in extracting metals from their ore, which is termed as bioleaching. Cellulose bacterium present in the rumen of cattle degrades cellulose. Certain luminescent bacteria emits light. Botoxin produced by Clostridium botulinum is used in the treatment to remove facial wrinkles. Mycoplasma, which are otherwise called as pleuropneumonia-like organisms, are the smallest known aerobic prokaryotes without cell wall. Mycoplasma have been reported to cause a number of plant and animal diseases, and these have also been reported from urinogenital tract of human beings. In human beings, Mycoplasma hominis causes pleuropneumonia, infertility in man, and inflammation in genitals. As Mycoplasma has no cell wall, they have no definite shape, that is, pleomorph. Hence, they are called as the jokers of microbiology. Self-assessment after each session is mandatory. To help you with that, a link of a Google form is attached in the description box. Try it out. Meet you soon in the continuing session. Thank you.